genetic engineering ultimately requires inserting particular genes into organisms, different organisms to where they were obtained from. So we're going to look at 3 ways that genes removed from one organism can be inserted into another. So the first step in this is, let's imagine that we've got a segment of DNA and we have used restriction enzymes to cut a chunk out. So this section here. So we've used 2 restriction enzymes, one to cut each side of a particular gene. So we've got this fragment of DNA which contains a gene that we're going to manipulate. Now one thing that we might want to do is take this gene and insert it into a bacterial plasmid. So bacteria have one big single loop chromosome, but they also have some smaller loops of DNA called plasmids, which might look a little bit like so. So what we can do is we can take a plasmid from a bacterial cell, and we can use, so I'll do it in green, we can use the same restriction enzymes that we use to cut out our target gene. I might write that down. This is going to be our, or I'll call it our gene of interest. So if we use restriction enzymes to remove our gene of interest, we're going to use those same 2 restriction enzymes to cut a hole in the plasmid that we've removed from bacterial cell. So I might label this as a bacteria as well. And here we've got plasmid. So the reason why we would use the same restriction enzymes to cut out the gene of interest and cut a hole in the plasmid is because they will cut with the same sticky ends, because they're obviously going to cut when they see the same sequence. So the next step in this process is we're now going to see a hole has been cut in our plasmid, which will then allow those sticky ends to come into play, and our original gene of interest is able to be inserted into our plasmid. So we can now call this recombinant DNA. So recombinant DNA is DNA that's a combination of DNA from 2 species as a result of genetic engineering. So we've got this plasmid now which has embedded in it the gene of interest that we might have removed from a different organism. Might have been a plant or something like that. Now, with this plasmid, what we can do is we can, that's pretty wonky, we can reinsert it into a bacterial cell. So we can have a couple of plasmids that might have been unchanged. Now we've got our recombinant plasmid that contains that gene of interest. So there's a couple of things that we might, a couple of reasons why we might have done this. The first reason is we might be trying to negate bacterial resistance. So we might have a strain of bacteria that is resistant to antibiotics of some description, and by adding a particular gene, it's going to mess with their ability to be resistant to this antibiotic. So we want to create a bacteria that doesn't have that resistance, allow it to breed and multiply, and we are then going to have a strain of bacteria that can't fight off antibiotic treatment like it originally could. What we could also want to happen is, and I'll draw a little graph here very quickly, if we look at time versus bacteria grow exponentially. So they divide very, very quickly, and their numbers will go up something like that if we allow them to divide over time. So it means that 1 bacteria will become 2, 2 will become 4, 4 will become 8, and they double every generation. So we can allow these bacteria to double, so eventually we are going to have hundreds of thousands, if not millions of bacteria, and we can remove, oops, I have to redraw that one. We can remove all these copies of the plasmids that will have copied and now contain our our genes of interest. So every bacteria that is a copy or a clone of that original one through binary fission is going to contain that gene of interest. We can then remove those with restriction enzymes, and we've got all these copies of that initial gene that we're able to do stuff with. So what we're able to do with these copies of a gene now is we can insert them into different organisms in different ways. And there's two kind of main ways that we can do that. So the first thing that we might want to do with this genetic material is we might want to use a viral vector to insert that genetic material into an organism and try and get it taken up into its genome so that we get a particular effect. So I'll stick with red. So let's say we've put a bit of that genetic material, so one of these segments of the gene into a virus, and then we're going to allow that virus, or introduce that virus to an organism where it's going to move into and ideally join to and infect a host cell. I'm drawing a bit of a nucleus here, and we will see the particular gene make its way into and join onto the genetic material already existing inside of a particular host cell being infected. So we can add a gene that has been genetically engineered, copied into a virus, get that virus to infect a host cell, and then that gene might be taken up inside of the genome and we'll get a particular effect inside of that cell. What we can also see is a micro-injection of genetic material into a fertilized cell. So we might have on my butt. What we might have is we might have a cell here, a zygote. So this is a fertilized cell. It is a combination of an egg cell and a sperm cell. And inside the nucleus here, we've got our genetic material. I'll just draw it as kind of a bundle of chromatin, say. So what we could do is we can remove this genetic material. We can remove this genetic material from a zygote. And we can then use a fine glass needle. So this is all going to be done at a microscopic level. But we can use a fine glass needle to inject our new, actually I'll stick with red, inject our new genetic material into this particular zygote. So we can take out the original DNA that would have come about varied because of the sperm cell and the egg cell, which would have combined. And we can then insert some desired genetic material into this zygote so that we get progeny the way that we are trying to, the way that we want, with the specific characteristics that we want. So this is called micro-injection. And this kind of thing has big applications in cloning, because what we can do is we can take this genetic material here. This genetic material may have come from tissue cells, or sometimes referred to as somatic cells, from an organism, and we then embed it into a zygote. And what's going to happen is that zygote obviously is the very first initial cell of an organism. Every division of that zygote from this point on is going to end up with that same set of DNA that we have obtained from elsewhere, different organisms. So this could be a clone. If we take tissue cell or somatic cell from another organism, this can create a clone because it's going to see the same copy of DNA. And we've had that occur with organisms as large as a sheep. A couple of decades ago, I think it was the late 90s, Dolly the sheep was cloned and it was viable for a period of time. It died a bit young, but there was a successful clone of a sheep made through this kind of process.